everyone. Hope you're enjoying the sessions so far today. Those were great stories from the families. So thanks so much to all three of our speakers for sharing with us. So now we're going to talk about searching for a cure with gene therapy. And I know there's a lot of excitement about this panel. We had our happy hours yesterday, 2 happy hours, and there were just a ton of questions about gene therapy and what it could mean for our kids and just people trying to understand it. So I'm really excited about this panel. We have 3 speakers today, one of whom I spoke to. He wasn't available today, so I spoke with him earlier in the week, and we'll share that conversation. But we have Amy Ramsey, who's going to talk later on. But we're going to start with Adi Barzel, who's going to kind of give us an overview of gene therapy. And Adi, can I let you introduce yourself?
Sure. Thank you very much. Can you hear me well? I'm excited and honored to be here with you today. I am a professor at Tel Aviv University, and also am the president of the Israeli Society for Gene and Cell Therapy. Previously at Stanford with Professor Mark Kay, and together we started a gene therapy company called Logic Biotherapeutics. And today I'll share with you some of the data which led at the of Logic Bio. And Logic Bio is interested in curing rare genetic diseases, mostly such that are concentrated to the liver. So not necessarily directly relevant to Grin. However, I think you will see some commonalities and many ideas that are shared and perhaps can be implemented also in future gene therapy of Grin disorders. So I will try to share my screen. Let's see if that works. So hopefully you can see my screen now. So we're going to discuss therapeutic gene targeting, and that would mean that we are inserting a desired gene for a missing function in the child into the genome, so into the recipe that tells the cell how to work and the body how to work. And importantly, I hope to convince you that we can do that, at least for some applications, without breaking the genome. We do not need to cut the genome in order to insert new genes into it, but at least for some diseases, we can do so in a different way that I will share with you today. And we're going to discuss how this could be done using a viral vector. So a viral vector is something that used to be a virus, so to infect people. However, we as a scientific community have learned how to engineer it so it does not longer infect us to cause disease, but can still be used as a vehicle. So we can use it to code genes to be inserted into cells and sometimes also into the genome. In particular, today I will discuss the use of adeno-associated viral vector, or in short, AAV. You can see it here at the top. Virus, a dependovirus, which means that it can only replicate in our cells when another virus is present to help it. And it's not a cause of a human disease. So we're using a very safe virus, and we are making it even safer by taking out the viral genes and replacing them with, which is the gene of interest that we want to insert into the genome, together with some control elements that would tell it when to be expressed, in what cells to be expressed, and so on. So it's, as I said, it's non-pathogenic. Most of us have been infected with the virus throughout our lives, so we feel very safe about using it. It's gutted, so the viral genes are taken out, the new genes are put in, and there are already approved therapeutics in Europe and in the United States using AAV, including for spinal muscular atrophy and Leber congenital amaurosis and more. And this is the particular design that I have invented during my years at Stanford, which has led to Logic Bio. And here we can see an AAV at the top, coding for a transgene, which is the gene we want to integrate into the genome. And it is flanked by sequences, genomic sequences, and this helps them to integrate into the desired segment of the genome. This process is called homologous recombination. And using this process, we can integrate the gene into a desired place, which is the albumin locus. And the albumin locus, the place in the genome coding for albumin, was chosen because albumin is expressed very highly in our livers. So highly that more than 50% of all the proteins in our blood are just albumin. And that means that if you are able, as I will show you, to put your gene of interest into the albumin locus, you can have the cell as a factory producing a lot of your desired gene. And I will show you that it allows for stable expression, which means that once it is in the genome, it will be expressed in the cell, and every cell that is divided, split from the original cell. It is very safe. It is specific. It goes to a specific tissue and goes into a specific place in the genome. And it's also very simple. It's a plug and play technology. So for different diseases, you just put a different gene in order to compensate for the different deficiency that the child might have. So the first example is going to be in hemophilia, which is a blood coagulation deficiency. It is caused by a mutation in a coagulation factor. We need it for our blood to clot, and it's called factor 9, or F9 for short. So kids without F9 can bleed to death if they are not treated. However, luckily, and they can get the protein by infusions. However, they have to take it every week of their life, time and time again. And we hope with gene therapy to replace that with a single therapy. And indeed, in mice, using the technology I described earlier, we can get the mice to have high and stable levels of the desired protein, factor 9, in their blood, whether we inject the mice when they are young or when they are adults. And importantly, the clotting time here at the bottom has normalized. So we have cured the mice from the bleeding diathesis using a single injection. Yet a very different disease is called Crigler-Najjar syndrome. So here, the children have a deficiency in a factor that is required in the liver in order to get rid of a toxic substance called bilirubin, which causes the yellow coloration, but also when in excess causes systemic problems and unfortunately also early death. However, we believe that with gene therapy providing the missing gene called UGT1A1, we will be able to help these children. Indeed, in mice that receive just one injection of a gene-right vector coding for the missing gene, we have lifelong, for the mouse, lifelong expression of the protein, and they survive very well. Moreover, their brain function, here checking cerebellar functions, are normalized, and the levels of the toxin in the blood are very low. Yet a third disease called Methylmalonic Acidemia, and actually we are now in clinical trials in children in this disease. Here the children are lacking an enzyme, a protein in the liver called MAT, which is short for methylmalonyl-CoA mutase. And without this protein, they cannot process many of the food ingredients, and they have toxins in their blood. We hope to be able to provide MAT to the liver of these children and allow them to get rid of these toxic acids. Indeed, we were able to show that we can rescue neonatal lethal models of this disease, so the mice do not die. They can strive and gain weight when provided the therapy, and the levels of the toxin, the acids in the blood, are very low. We look now at the liver of these mice, and we can see evidence that the original cells that were corrected can now take over the liver instead of the dying cells. So step by step, the child has a more and more healthy liver. And we know that this growth is a healthy growth and not cancerous growth, because when we treat a healthy mouse, we do not see this growth. So this growth of the corrected cells is only in the situation where the surrounding cells are dying from the disease, whereas the corrected cells can thrive. So a final disease that I will discuss, and has the approach here has some relevance perhaps for future therapies also in Grin. So this disease here is called AAT, alpha-1 antitrypsin deficiency. And here the children, actually many of the manifestations come at later ages in the lungs because of damage caused to the lungs by elastase. This damage is usually prevented by AAT, the missing gene. So if the gene is not there, one has emphysema in the lungs. Another problem is cirrhosis of the liver, damage to the liver, because the mutated gene, the bad gene, causes harm to the liver. So we wanted to address both the lung problem and the liver problem. And we did so by providing the correct gene. This will help the lungs, but also an shRNA. And the goal of this shRNA is to knock down, to make much lower the expression of the bad gene in the liver. You can see both the positive effect of having more and more of the good gene to help the child or the adult in the lung, but also less and less of the bad gene and repopulation of the liver with the corrected cells. Okay, so with that I will conclude. I think I was able to show that this new technology called GeneRight is very broadly applicable, and we are now using it both to coagulation deficiencies as well as to metabolic disorders in the liver, such as Crigler-Najjar, Methylmalonic Acidemia, and in diseases like AAT, where there is also a problem with the mutated bad allele, bad version of the gene. We can provide the correct version while taking down the bad, faulty version. Okay, so with that I conclude. Let me stop sharing.
Thank you so much, Dr. Barzel. That was terrific. You're doing amazing work there. Because I know that there's a lot of parents on the call who have different levels of understanding. I just want to go back and just try and unpack a bit of what you talked about. So you talked about the vectors and how they are made from vaccines. So is it right to kind of think about you have the medicine that goes and gives the instructions of how to make changes in the DNA or how the DNA gets expressed, but then you also need a way to deliver that medicine to enough cells, and the vector or virus, that's how you deliver it? That's kind of how you pack the medicine?
Yes, so the reason we use viral vectors, so things that used to be viruses, is because viruses are so good in getting into our cells that for millions or billions of years to do. So we are using this great capacity of viruses, but we are avoiding the bad, features that viruses sometimes have. Some viruses cause diseases, and we would not like that to happen. So we take out the parts of the viruses that cause harm, but we leave the outer shell that helps the virus get into, also into the nucleus, the part of the cell where the action is going. And we are able to use this feature to bring the genes that are of interest to us, that help us cure the diseases.
Right. And so I think a lot of people have heard of CRISPR or gene editing, but that's kind of a newer and in some cases maybe potentially better kind of gene therapy. But that's not quite what you're talking about and what we're going to be talking about today as it applies to Grins. So can you kind of help us understand the difference between those a little bit?
Sure. So CRISPR is a fantastic new technology, and it has many applications, both in therapeutics and outside of therapeutics. And it allows us to cut the genome in a desired location. And cutting the genome can sometimes be helpful. For example, if you'd like to stop a gene from being expressed, right? You'd like to eliminate it. You can cut it and eliminate it. If you'd like to insert a very desired change, so you may cut the genome and also provide additional goodies that help at that location. So CRISPR is fantastic. However, it also has some disadvantages, and it's not the solution for all problems. Mind is that when you use CRISPR to cut the genome, there are also undesired side effects. So if you can avoid them, and that is what we were able to do with the GeneRight technology, then you're better off. And these side effects might include chromosomes, these are the parts of the genome, not being repaired the way you would like them to be repaired, sometimes remain cut, sometimes joined to other parts of the genome that are irrelevant or even dangerous. So sometimes it's better to avoid these consequences. One possible use of CRISPR is to try and hit the very mutation that each patient might have. However, this use of CRISPR, I think, is not necessarily the way to go with Grin and also with many other genetic disorders. And the reason is that very often each patient has a different mutation. And you not necessarily want to design a new drug for each and every patient, a new CRISPR, if you'd like, for each and every patient. It may be better, and I think this would be what my suggestion, to try and make a one drug fit all, at least fit all patients with a particular Grin 1 or Grin 2A, so for each gene, and one drug for each gene. And then you can try to insert the entire correct gene at a desired location. You may do it with CRISPR, but even then, I'm not sure that I would suggest doing it with CRISPR. It's in neurons, which are one of the main issues in Grin, you may not need to insert it into the genome at all. It may be sufficient just to bring it into the cell, and it will be expressed. So I wouldn't necessarily think that CRISPR is the first place to go for Grin.
Yeah, that's really interesting, because I think 90% of Grins and the broader Gris are single point mutations. So I think early on, I and other parents were really attracted to the idea of base editing, which is one kind of CRISPR where they basically just replace that single letter. But then, as you say, you need to make a different medicine for each variant, and that would be expensive and time consuming, where this other kind of simplified gene therapy, potentially there could be just one medicine for each of the genes. So yeah, that's fantastic. And then one more question before we go to the next one. There's kind of two different ways that these kind of gene therapies can work, right? So if people can think back to high school biology, the DNA is like the textbook that tells our cells what to do, or I guess the instructions, and then that the RNA transfers that into the actual directions. I don't know if that's an oversimplification, but you can target one or the other with gene therapy, and each has pros and cons.
So if I understand correctly, you're asking about targeting the DNA or the RNA? Is that
That's
the
right.
question?
That's right, yeah.
Yeah, so you would want the effect to be long-lasting. And for the effect to be long-lasting, it may be best to provide the gene as DNA. To provide a more transient copy of the gene in the form of RNA, but then you would have to provide it time and time again. So RNA therapeutics can be great. They can give you the desired effect. However, they are not once and for all. You'll have to give them time and time again.
Right. So I guess some people might think it's an advantage because it's not permanent necessarily what happens with RNA, but yeah, the drawback is you have to do it time and time again.
I mean, it's a question of resources. I think it's an interesting. I wouldn't rule it out.
Yeah.
I think AAV would be my first choice if possible. Maybe more challenging for some green, easier for other greens. But RNA time and time again is still a viable option, and definitely something I would look into.
Actually, I do have one more question before we move on, which is, you talked about all this amazing work that your team has done on mice. Can you talk about some of the successes that we've already seen with gene therapies in people?
Yes, so there are some approved drugs, and also successes in clinical trials, which will lead to approval by the FDA, hopefully in the coming year. So approved drugs we already have in Leber congenital retinal deficiency, so children who are losing their eyesight, but this deterioration can be stopped, and in some cases perhaps even to some extent reversed, using gene therapy based on AAV that goes into the retina and provides a missing gene. And then there is a gene therapy for SMA, spinal muscular atrophy, and these children have a problem in the way the brain sends the orders to the muscles. So in the junction between the neuron and the muscle, they are missing some gene function there. And again, it can be provided by an AAV, a lot of AAV given systemically, so it has to go to many muscles and many junctions between neurons. But if the child is diagnosed early enough, then there are amazing effects. So it's very approach. There are also gene therapies using hematopoietic stem cells, and many newly approved gene therapies for cancer, where the engineering is done to immune cells, allowing the immune cells to fight the cancer in a better way. So to several types of cancers of the blood, we now have very potent gene therapies. And many are coming. I think in the coming year, we'll see approvals in hemophilia. Exciting times.
Okay, thank you so much for that. And we will now turn it over to Dr. Albert Seymour from Homology Medicines.
Albert, great to talk to you. Thanks for joining us at the Grin virtual conference. Can you start by introducing yourself and telling us a little bit about what you do?
Yeah, sure, Keith. Hi. First off, thank you for inviting me to the conference. I'm, always a fan and enjoy talking and discussing with you and about the Grin. So my name is Albert Seymour. I'm the CSO of Homology Medicines. Just real quick, I'm a human geneticist by training, and then from a industry perspective, have had jobs at Pfizer, was there for almost 15 years. Shire Human Genetic Therapies was there as the head of research and non-clinical development, and since 2016 have been here at Homology working on developing the AAVs that we have as part of our technology platform.
So what's Homology's approach to gene therapy medicine?
Yeah, so Homology was really formed and founded based on the discovery of 15 what we call adeno-associated viruses. So these are called AAVs. And the 15 that really formed the company were discovered in human hematopoietic stem cells or human bone marrow cells. And that's why we refer to them as AAV HSCs. And we like this set of AAVs because A, they're naturally derived. So they were found in healthy individuals. So we know that they can reside within healthy individuals and not really cause any pathology or any kind of disease. We've then taken these 15, and what we've been able to determine with them is that when we inject them into non-human primates or mice, we see that they go to a variety of different tissues. And this can be the liver, this can be the muscle, but more importantly for Grin in particular, we can cross the blood-brain barrier and we get into cells that are relevant within the brain as well as in the spinal cord. And so as we were developing and characterizing these, we really found that these AAVs could be a workhorse or drug delivery approach for Homology Medicines. And since then, over the past 5 years, we've taken these AAVs and used them to deliver genes that are missing. We have one program in the clinic right now for a disease called phenylketonuria or PKU. And we have another program that's coming to the clinic soon for a disease called Hunter syndrome. And this is a rare lysosomal storage disorder. And what we do is we can engineer the inside of the AAV to deliver to the cells in the body a gene that's missing. And that's a similar approach that we'll be taking with you and the Grin studies with Amy.
That's so exciting. Okay, so we first met, I guess, a couple of years ago before COVID. Laura and I came down, and you and the CEO Arthur were gracious to host us there.
Yeah, I remember that like it was yesterday, and it's funny because that was actually in a different building that we're in. And I remember you talking about Grin and your son Bryson, and I remember going through all of that and thinking, you know, if there's anything that we could do to help out with that, that we would really be willing and eager to help out with that.
Yeah, and so we asked at the time, I think, you know, we were learning about your vectors and how you thought they were better than some of the others out there at delivering to the brain, as you mentioned. And so, you know, we asked at some point when we're ready, would we be able to potentially use those to test in Grin mice? And you guys said yes way back then, and now that's finally going to happen. So can you tell me about the medicines that you're making for the Grin mice?
Yeah, absolutely. So we're working very closely with Dr. Amy Ramsey. And so the mice that are available, we have two types of mice. One that both have mutations in Grin1, one that has an activating mutation in Grin1, and one that has a loss of function mutation in Grin1. And what we're doing is we're working with Amy to clone in the wild type or normal gene for Grin1 into our AAVs. And what we'll do is we'll use those AAVs to deliver with 2 routes of administration. We're looking at trying to target the relevant regions in the brain and the spinal cord with either an IV, intravenous administration, but we're also going to look at going more direct into the spinal cord, into the CSF, which is a route of administration called intrathecal. So we'll go into the intrathecal space. And what we're doing is we're developing those medicines. We're using one of our AAVs that crosses the blood-brain barrier and gets into many different regions of the brain and spinal cord at very good levels, both in the mouse as well as in non-human primates. And what we're going to do is we're going to use those AAVs to deliver this normal copy of Grin to as many cells as we can. And the way we do this, we design this, is that we use what's called a promoter that sits in front of that gene. And that promoter is kind of a signal to the cell that once the virus delivers the vector genome to the cell and gets into the nucleus of the cell, the nucleus in the cell itself will recognize that promoter and turn on the transcription and ultimately make the protein Grin1. And then the cell naturally will assemble and utilize and move Grin1 to where it needs to be. And so that's the approach that we're taking. So we're making a normal copy of Grin1, we call it a wild type, that we can use these AAVs to deliver to different cells in the body.
So Grin disorder is almost always heterogeneous. So patients will have one kind of good copy and one broken copy, I guess, of the gene. And so what you're doing is is it kind of like you're adding a third copy? So they end up having two good genes, like one natural one, one synthetic one, and those will hopefully water down the bad genes if those proteins get made, or replace them if those proteins don't get made?
Yep, absolutely. And that's really the focus of the research that we're doing with Dr. Ramsey. So we're taking our medicines, we're going to be administering them into the mouse model, and then testing that exact hypothesis. So we're not going to change the mutation that's already there in the mouse. We're going to add a number of copies of a good copy of the wild type normal copy and see whether or not we can just in a sense dilute out the effect of the mutation that currently resides in there. So yeah, I guess that's a neat way to look at it. You'll have the mutated copy, another copy in the mouse, and then we're adding a third copy, which will be it's actually the human Grin1 that we're going to overexpress in the mouse.
Okay, and I think it's almost the same. Grin1 is a really well preserved gene.
Very well
conserved.
All
of the Grins are very well preserved. Yeah.
Okay, and I think you mentioned two Grin1 variants. I think there's also, hopefully there's plans for a second phase that would include a Grin2b mouse as well. So just because I know there's a lot of Grin2b families on the call too.
Yes.
And so I think one of the challenges with, other than the Grin1 is a medium-sized gene, Grin2a and Grin2b and Grin2d are larger genes. And I think with some types of vectors, that's a challenge to deliver these larger genes. How do they work with the vectors that you guys use?
Yeah. So within the AAVs that we're using, they typically have a capacity, meaning the amount of synthetic DNA that we can put into the viral genome, typically around close to 5,000 base pairs. Grin1 is close to 3,000. I think the exact is 2,830 base pairs. That's a great size because that's the size of the cDNA that's the instructions to make the protein. We then have to put a promoter on the front of that. So as we go larger with the cDNA, so things like Grin2 that are probably closer to 4.7 kb, what we'll have to look at is whether or not we can use very small promoters to still package it within the AAV confines. It's not impossible, but it certainly will involve a little bit more engineering and testing to identify a small enough promoter that can still express that large of a gene.
Right. Okay. And one of the things that we talked about when we were building our scientific plan at Cure Grin is, the idea that it would be optimal if there could be just one medicine that's created for one gene, like as opposed to CRISPR or some other medicines where you basically have to make a different medicine for every genetic variant, and there's so many variants with our kids. And so one of the ideas that we talked about is that potentially the best option would be something that both does what you're talking about, where you're kind of adding a third gene, but also knocks down the wild type gene. Is that something that you guys do for any of the diseases that you're looking at, or?
We haven't to date, but it's a very good question, and it's certainly a possibility within AAV. And so because you have so much flexibility around what you put into the AAV to deliver, there are examples where you can deliver to express, say, a normal copy of Grin1. And then maybe downstream of that, you have the same vector express maybe an siRNA or something that would target the mutated form and knock that down in the cell. So it is certainly possible. And I know others have looked into approaches like that. And even internally at Homology, we've actually considered some of those for some of our other programs where we do want to reduce the amount of the mutated copy while still overexpressing the good copy that we put into there. So.
Okay. This has been great. Is there anything else that is important to talk about in terms of this process? We're going to hand things over to Amy Ramsey now to talk about what she's doing. But before we do that, anything else to mention?
Yeah, no, I think it's just, the collaboration has been going along fantastic. And, we're looking forward to making these medicines in AAV and getting them into Dr. Ramsey's hands so that we can then test, and do the research around, is this a viable approach or not? I think we'll know the answer to that soon. And so time will tell. We look forward to when we get the results from these initial set of studies.
Albert, thanks so much for joining me
Yeah.
and joining our conference. Thank you.
Yeah, absolutely. Thanks, Keith.
So Amy, this is something you and I have kind of known was coming for a year, and it's exciting to finally be able to talk about it. This is huge news.
It is. I'm humbled to be part of it. I hope we can deliver some good news.
Yeah. So I know you have some slides to share, but I wonder, are you going to be talking about, because I thought it would be useful to, just as background, to talk just quickly about your previous work that on the rescue mouse.
Sure. Right. Yeah, I think that turns out to be a really important question for gene therapy, and we shouldn't take it for granted. So the question is, with a neurodevelopmental disorder, will we be able to provide a gene in a child or even in an adult that will be able to treat the symptoms? Or is this something that really we need to go very early on, in the child's life when they're still an infant? So the work that we did, that we published last year, was to show with a genetic model with mice that we could let the mouse grow up with very few NMDA receptors, and then use our little mouse genetic technology to engineer the gene and recover the normal form of the gene. And then we could test to see whether those mice had improvements in their symptoms. And the really exciting thing was that, yes, we were. So I think I have a slide that I could show you that just kind of does a quick overview of what it looked like. So just to give you an idea, this was from this paper where we. You can see up in the top left the levels of the mRNA of the gene expression for Grin1 in the mouse normally. And then in our model, they have very low levels of Grin1, which is that middle left panel. And then on the bottom, you can see the level of recovery that we got with our genetic trick. So we definitely improved the amount of Grin1 that was being made, but we didn't restore it back to 100%. And despite this, we still had very good recovery in terms of their cognitive abilities and really just their health. They gained weight. There was a lot of things about these mice that they seemed better. And importantly, we didn't see any of their phenotypes worsen. So I think that's another thing, is we weren't sure ahead of time whether we would see more seizures or we would see something new that would be a problem. So that was really encouraging for us to know that, yes, indeed, the Grin disorders seem to be amenable to gene therapy. I don't believe the same things have been done yet for GRIA or for GRIC, but I think that it's certainly worth exploring and working on these therapies for those disorders as well. There's no reason to think that they wouldn't be similarly amenable to gene therapy.
Right. And so we'll learn, even more about that, I guess, in this next phase of testing. So once you get the medicines from Homology, what happens next?
Okay, so I'll go back to sharing my screen so you can see our plan. So these are the things that we're hoping to answer with these studies that we're going to be doing. And I think Albert talked about this a little bit. So we're going to see what's the best route of administration, because we're going to try IV, and that will go everywhere throughout the body and throughout the brain. The brain gets, a lot of nutrition from, a lot of vascularization. So the virus will certainly go there, and they have these protein capsids that are able to go into the brain and deliver those instructions into the brain. So that would be nice if we can achieve that, because it's not very invasive. But the other way that we'll try and do this is to do into the cerebrospinal fluid into the spinal cord. So, this is a little bit more invasive, but really much less invasive than doing something like intracerebroventricular, which is where we would make a hole in the skull and deliver the virus directly into the brain. So I'm very excited to try these. We're also going to try a couple of different promoters, and I'll show you what I mean there. So then once we try all of these different combinations, we'll be looking to see how much new Grin1 or Grin2b the brain is making, what types of their phenotypes are improved, whether we see any problematic phenotypes. So do we see new things happening that we don't want to happen? And then we also want to test it in a bunch of different models, because it's kind of easy to see how it could help if you have a loss of function, that by supplying another copy, you would be supplementing a lack of the receptor. However, because a lot of these variants they're kind of acting like a poison subunit, where their one bad copy can interrupt the function of the whole receptor, which is made of 4 subunits. We actually think that we could use the same strategy even for gain of function by, as you say, diluting out that poison subunit and providing more that's the healthy subunit. So this is what we're doing. We're going to try this synapsin 1 promoter, which will really make just the neurons produce the Grin1. We'll also try a different promoter called GAPDH, which should express in all types of cells. And then later on, we're going to try and put the Grin2b instructions behind a very small minimal promoter that's only 500 base pairs. So we are going to see if we can fit this whole thing into the AAV so that we could try it in the mouse model. So Homology is going to prepare the viruses. We've sent them the constructs, and they're now getting started on making the viruses. They'll send them back to us. And essentially these viruses are just delivery vehicles for the instructions. And as Dr. Barzel said, the way we have ours designed, they're not even going to integrate into the DNA. They'll just be hanging out there as their own little fragment of DNA that can be used to make the Grin1 mRNA and make more of the Grin1 protein. And then what we'll do is we'll try that, as Albert mentioned, we're going to try that on the loss of function variant. We'll try it on a gain of function variant. And then we'll be making this loss of function variant of the Grin2b gene, where we already know that a lot of clinical data around the patient who carries this variant, and we'll be making the mouse that carries that as well. So we'll look at behavior, we'll look at how their NMDA receptors are functioning, we'll do lots of histology of the brain and other parts of the body to see where is the virus going, how much of the viral payload is being expressed and turned into mRNA and into protein. And another thing that's important is to see how long does this last. So we'll set some of these mice aside and let them age. And we'll do the same thing to see, is this a persistent benefit that we would get with these experiments? So I think those are the main slides that I have some others if you want to see them, but I hope that kind of conveys the scope of what we're trying to do in these first studies.
And so if we do see success in the mice, what happens after?
Well, I think that Dr. Barzel can probably answer that better than me, because I'm really not an expert in the pharmaceutical process of this, right? And the regulatory steps in this. So I for other drugs, and I assume it's pretty similar for gene therapy. There's a lot of toxicity that needs to be done in multiple types of animals. You need to show that it's safe. And then you would start, after you've shown that it's safe, you would make an application to the FDA or whatever, in whatever country you're going to do the first clinical trial, you would make an application to that agency to do the clinical trial. And then they would give you the permission to try it first on a small number of people. And then if it's safe in that small number of people, they would let you do it in a larger population. The timeline for that, I think Dr. Barzel could probably answer that better than me.
Yes, I can say that we've gotten the first funding to start the company in 2016, and we are in clinical trials in 2021. Range. So it's not a short trip by any means. I do hope that from one gene therapy to the next, as the regulator and the community in large become more and more familiar with this type of technologies, especially now with the COVID vaccines, which are different types. There are several flavors here, but different types that the regulator and the community are feeling better with genetic medicines. It will take a shorter timeline, but still it's a journey and something that we all have.
Right. This is so exciting. So I want to open it up for questions from the audience, but for some reason I can no longer see my chat or Q&A feature on the right side of my screen. Amy or Adi, can you see yours?
The last that I see is Amala's question about whether AAVs can be used for all GRI genes, and I think that we'll try that eventually. I think that would be my hypothesis going forward, is yes, but we will see.
And so, so as I said, I can't see it, but if Amy's able to see it, there's two different areas there on the screen. One is for chat and one's for Q&A. You can put your question in either one, and we'll watch, or Amy will watch for it.
Yeah.
Yeah, so one question here is, do you know which locus you could insert the GRI genes or shRNA into, or how you would deliver AAVs to organs other than the liver? So with regards to different AAVs, I think that the approach taken by Homology is promising, and I think you're doing great to have Professor Ramsey collaborating with Homology, and I'm hopeful that these AAVs will reach the brain. They are slightly different than the AAVs that I used to go to the liver, but still there are commonalities, and I think that's promising. I don't think you need to go into a specific locus in the genome. That was important in the liver.
I think Adi's freezing up a little bit.
Yeah, he is freezing up. I think what he was going to say is the cells are dividing. Oh, there he is.
Have I lost you?
You were frozen for a moment, but we can hear you again.
Okay, great. So I was saying that I don't think you necessarily have to of neurons. The approach taken by Professor Ramsey, I think, makes perfect sense, just to try to express the missing genes from the AAV as a separate molecule, not integrated in the genome. It may be necessary to use shRNAs in some instances. It's too soon to know. So I think it makes sense to first try just to overexpress, to make a lot of the correct gene. And then in those cases where it seems that the mutated gene is toxic and expressing the correct version is not sufficient, then one may think about also expressing an shRNA to knock down the expression of the mutated allele. In itself, it's very feasible. The challenge these all point to is the coding capacity. So how much can the AAV fit in? So this relates to the previous question of different Grin versions that are of different sizes, and the larger you are, the more difficult it is to put it in an AAV. And if you would shRNA, the capacity becomes challenging, but perhaps still doable. I would definitely go and try it.
Keith, it looks like there is a question here about what are the sources of funding for the research, which is always an issue for scientists to find that funding. So actually, Grin has been really great about helping to identify those sources of funding. I think some of the most powerful funding actually comes from government agencies. So governments are able to provide funds that just charities are not able to. You know, just the amount of resources that governments have is so much more. Having said that, there are some charities that are supporting this, and the Simons Foundation has one of their focuses is autism, and some of the Grin genes have been identified as autism susceptibility genes. So with Grin, you know, I have been applying with Graham Collingridge, who is hopefully also here, and he's the one who made the G620R mouse that we're going to be using for some of these gene therapy trials. But anyway, we put together a grant with Homology Medicine, a grant proposal that we're asking them to help us fund this. But at this point, we're funding it through, you know, sources that we cobbled together from government and private foundations like Grin.
And the hope would be that, you know, that this is kind of the proof of concept, and that after that, Homology or another company will see the proof of concept, and then, you know, for when it gets to human trials, that would typically be funded by the biotech.
Yes, I think Dr. Barzel, Adi, you mentioned that, and you know, when we were doing some of our planning about what Grin should focus their efforts on, I think you mentioned that what we really need to do is de-risk this so that it becomes palatable for a company to take this on.
Definitely. In the end, it has to be a company. Taking a drug to the market costs dollars. So there is no other source but the free market. Somebody has to make money. So we need to de-risk it so that investors would put money into company that will take it further. But that's what we do in the academic setting. We do the first experiments to showcase that it is possible, and then raise the interest of biotech.
Are there any other questions?
There's a question here. Let's see. I thought that brain cells are very hard to target with AAVs. So would it be hard to reach enough brain cells? Is this a concern or something you will test? Yes, that is the big concern. So looking at what is the level of expression that we're getting. One thing that's encouraging is from our earlier studies where I showed you that picture with the orange brains, is that we don't need to get a huge amount of expression to make a big difference in the symptoms. So we'll just have to find out, first of all, how effective we are at delivering that message, and then how much, you know, do we actually need in order to get the benefits that we're looking for. So Do you have more to add for that, Adi?
No, I'm not myself, have not tried gene therapies in the brain, so I completely follow you.
Right. So I think that, speaking with the scientists at Homology, they are focusing on brain disorders, and they have identified those vectors that are really good at getting into the brain. So I think that of all of the ones that they've developed, they're focusing on the ones that they know will work at getting into the brain. And even you can choose ones that are better at getting into certain cells within the brain. So some of them are really good at getting into neurons. Some of them are really good at getting into astrocytes. We're really not sure what is going to be the best route. So we need to try a lot of different variations at this point.
We've got about 4 minutes left, so we have time maybe for a couple more questions.
Is there a conceptual workaround available for the AAV load limit?
So there are approaches being taken. None of them is trivial or proven in the clinic, but there are early proofs in mice that one can deliver 2 AAVs, each coding for half of the protein, coded in a way that will allow the 2 halves to become a functional version inside the cell. This is less of 1 AAV, but in some cases, there might not be any other choice. And at least in mice, in some cases, this has been established. So yes, this would be the workaround if we are not able to put larger genes all at once in 1 AAV.
And I wonder if, we talked we talked before about the knockdown and replace idea, and definitely with the current AAVs, you couldn't fit a knockdown in with the Grin 2 genes, but potentially if you could add the knockdown, then that could help make up for the fact that you're splitting what you lose by splitting into 2.
Potentially.
There's a question here. I think this is about the work that we did, the kind of foundational work we did. Did adults rescued respond differently, equally as well as young mice? So actually, we did see a better improvement when we did this same rescue in young mice. So juvenile mice, we saw a more complete recovery of the many different diverse brain functions. However, we were really impressed at how much we saw even in the adults. So for the mice, it didn't make that big of a difference. We'll see humans are different than mice. So, I can't say that would be true, but I think that eventually, yes, we will get a better outcome the sooner we intervene.
Well, I want to thank Dr. Ramsey and Dr. Barzel for joining us here today. This has been really great, and Dr. Seymour as well. Thanks for doing this. I think that our community probably has a much better understanding of gene therapy now, as well as a lot of excitement about what's coming. So thanks very much. So I just want to let everyone know that we're taking a 15-minute break after this, and then when we come back, we're no longer on the main stage. So there's actually 4 different sessions that you can choose from. So on the left side of your screen, there's one that I think says breakouts, and there's 2 different sessions to choose from on there. There'll be another button that talks about language and youth session. So in this next time, there is both and I don't have the schedule in front of me, but there's a language session, and there's also the sibling session. So yeah, so lots to choose from. You get a 15-minute break. Go get yourself a coffee, and we'll see you back at 1:15 Eastern. Thanks, everyone.
Thank you very much.
Bye.